Good evening, guys. Hope you all can hear properly. Okay, guys. So let's move on with yet another chapter, that is cell cycle and cell division. So we are having the first lecture. I hope to complete this chapter by two lectures. First lecture with mitosis and second one with meiosis. So let's move ahead and complete the first lecture of mitosis. Before that, if anyone new is here, just do not forget to join the Telegram group t.me/biopointnew, where we conduct the regular discussion, daily quizzes, questions, updates on classes, and OMR exams. And guys, just join our Telegram group to know more about the crash course for Neet 2021. Once again, reminding you, do not forget to hit the like button. Do share it with your friends. Do not forget, if you're a new member, do not forget to subscribe our YouTube channel BioPoint, stream the world of bio, and do not forget to leave a comment as a remark. So guys, let's start with today's lecture on cell cycle and cell division. We are having amazing slides today waiting for you in this chapter of cell cycle and cell division. Hope it's perfect and fine. Today, hope it's perfect and fine. I don't know why it's blurred now. Is the screen blurred? Are you able to read whatever it's there given there on the screen? Can you read whatever given in the screen? Okay, Harshita. Okay, if it is not adjustable, just kindly inform me by using the comment section. And anyway, we are going to start today's session on cell cycle and cell division. Just everyone, just refresh. Just refresh. Okay, then. We are moving on to the chapter cell cycle and cell division. Let's see what's awaited there in this chapter. So all of you hearing the name cell cycle, what do you mean by the term cell cycle? What do you mean by cell cycle? Yes, exactly what you all mean. Cell cycle refers to the life cycle of a cell, right? The life cycle of a cell is known as the cell cycle. And what else? If we there, just wait, please wait. Please wait. One minute. Okay, so cell cycle refers to the life cycle of a cell, the life period during which a cell, during life period of a cell during which a cell synthesis DNA replication, synthesis DNA grows in size and divides into two daughter cells. It's bit getting lag because it's given I like this in 3 lines. Okay, that's fine. Just leave that. Anyway, we are gonna do it. We are gonna start it. Okay, just leave everything. We are going to start now. Okay. Cell cycle. So what is cell cycle? It is a life cycle of a cell. the sequential pathway of a cell that divides finally into two daughter cells. A single a single cell just divides into two daughter cells. That is what we say to be. That's what the base of this cell cycle. Okay. So duration of cell cycle varies in each organism and each cell type. Duration of typical eukaryotic cell cycle is about 24 hours. That all are important. That all are neat questions. Duration of a human cell, the cell cycle of a human cell, it is 24 hours, and whereas it is in case of yeast, it is 90 minutes. All those we will learn. Okay, so before moving on to that, we have to discuss why the cell cycle is important. What happens if a cell doesn't show cell cycle, and all those things are the topics that we have to say as an introductory part of this chapter. Clear with that? So before moving on to the what chapter, that is a further division of a cell, let me say what happens if an organism lacks cell cycle, what is the importance of cell cycle and all those things. Clear with that? Are you okay? So first point that you should add, what happens if a cell doesn't show cell cycle? What happens if a cell doesn't show cell cycle? If a cell doesn't show cell cycle, it leads to chromosomal abnormality. Leads to chromosomal abnormality. Chromosomal abnormality. Okay, that's why cell cycle is important, because it leads to, if cell cycle doesn't occur, it leads to chromosomal abnormality. Then, so that's we discussed why what happens if a cell doesn't show cell cycle. Then what is the importance of cell cycle? It is to maintain the nucleocytoplasmic ratio, the NC ratio that we will learn at the what further topics of this chapter. Okay, so the NC ratio, in order to maintain the NC ratio, we have this cell cycle, that is the nucleocytoplasmic ratio. Clear with that? Is it clear? Hope it's fine. Okay, then we are moving on to the further topics. The cell cycle and cell division. What is cell cycle? We have discussed that is the life period of a cell during which. One second. One second. I will come now. Just wait. I'm just searching why this is blur. That's why. Okay, I'm back. Okay, so as you all know, the cell cycle consists of two phases. Okay, from now onwards, we will be moving in a flow, and I will not be going on to check all those things from now. So just focus. Sorry for disturbing. So the life period of a cell during which a cell synthesis DNA grows and divides into two daughter cells is known as the cell cycle. Clear with that? Okay. So a cell cycle has two basic phases. A cell cycle has two basic phases. That is, one is the interphase, and the second one is the mitotic phase. Mitotic phase. And guys, Animal Kingdom will also be completed. It got a little bit lag, but soon it will be completed. Okay, mitotic phase or the M phase. So the two basic phases of this cell cycle is the interphase and the mitotic phase. Mitotic phase or M phase. Okay, mitotic phase or M phase. Okay, leave that. Let me use the pen of this PowerPoint. Let me check whether it will be okay or not. Okay. Then we're going to learn in detail about this mitotic phase and interphase, and this lecture we are going to have about this mitotic phase. So interphase, it is the phase between two successive M phases. That all things you will understand. Before that, just wait and read these statements. Okay. And mitotic phase represents the actual cell division. So we should know about more about this interphase and mitotic phase. So let's learn about that. Okay. So all of you know that interphase, it is the resting phase, right? Interphase is known as the resting phase. Interphase is known as the resting phase, and it is the longest phase of a cell cycle. It is the longest phase of a cell cycle. That is, two phases are there, my M phase and interphase. Among that, interphase is the longest phase. M phase we have M phase, that is the mitotic phase. Mitotic phase, and this mitotic phase is the shortest phase. That is, in a cell cycle, about 95% it is interphase, and only the rest 5% it will be the M phase, that is the mitotic phase. Clear with that? Okay. Now, then duration of cell cycle. Duration of cell cycle, if we say about that, the embryonic cell, all these are important questions and previous year need questions. Okay, so the embryonic cell has a duration of 30 minutes. The cell cycle of an embryonic cell has a duration of 30 minutes. Yeast, as we have discussed, guys comment me the cell duration of cell cycle for yeast as well as the duration of cell cycle for human cell. Yeast and human cell, comment me right now. Everyone, if you are here, give me thumbs up too on the screen. Comment me fast. And then stomach cells, if we discuss about in case of the stomach cells, what we have to say is every 3 days. Oh my god, I'm not able to write. Every 3 days, this stomach cells will be replaced. Every 3 days, this stomach cells will be replaced because it is there. Yes, Harshid, and 24 hours in human cells and 90 minutes in yeast. Okay, exactly, Calvin. Yes, Akash. Yes, hello. Okay, so this is about the duration of cell cycle in certain cells. Clear with that? Next, we are gonna learn in detail about this interface. Okay, then interface, it is the phase between two mitotic phases. That is, how the interface goes is mitotic phase. After mitotic phase, you have a resting phase. That is known as interphase, and after that again comes the mitotic phase. So this G1 phase, S phase and G2 phase, these are the different stages or the events of this interphase. Okay, certain events of the interphase is G1 phase, S phase and G2 phase. Okay, these are certain phases of this interphase, whereas this mitotic phase or the M phase consists of two main phases, that is the karyokinesis and the cytokinesis. Karyokinesis and the cytokinesis. Clear with that? So karyokinesis refers to the division of the nucleus and cytokinesis refers to the division of cytoplasm. As per the term karyokinesis, karyo refers to nucleus and cyto refers to cytoplasm. So division of nucleus and division of cytoplasm. Clear with that? Is it clear till now? Give me thumbs up on the screen. All the seven present. Fast. So its M phase is starting there. Before that, we have to learn something more about this interface. Okay, guys. Then interface. No division of cell occurs in case of this interface. No cell division. I don't know why I'm not able to write now. No cell division in case of this interface. It is known as the resting phase. As all of you know, it is known as the resting phase. So why it is known as the resting phase? Because of the absence of cell division. Since no cell division is present, it is known as the resting phase. Clear with that? That's why it is known as the resting phase. And another point coming is for your confusion, it is the most active phase of cell cycle. In the same way, the interphase is the most active phase of cell cycle. Most active phase. Why it is called so? Why interphase is called so? Anyone, if you know, comment me the answers right now. So till then, I will be discussing some other points. So if anyone know why it is known as the most active phase of cell cycle, comment me right now. So the interphase, it is known as the resting phase because no cell division, no division of cell occurs, and what happens instead there? Instead of no cell division, what happens? What happens? Changes in nucleus occur, right? Nuclear content changes in content of nucleus. Okay, if it is containing 2C chrom, that all we will learn. Okay, that all we will learn. So before don't get confused with that. After learning meiosis, we will discuss all those things. Yes, exactly, Harshita. It is known as the most active phase because maximum cell metabolism and cell growth takes place. Maximum cell metabolism and cell growth take place. Clear with that? That's about interphase, and interphase has three subphases. That is the G1 phase, the G2S phase, and the G2 phase. We will learn in detail about all those things now. So till then, please wait. Clear till now? Tell me the truth whether any part is confusing or anything it's not clear. Is it clear till now, guys? Let me search the slides by that time. Just G1 phase is facing phase there. Okay. So we have discussed in case of what are base points about this interface, and now we are going to discuss some of the base points of. Okay, Harshida, Nila, and Calvin. Very clear. Oh, sorry, very thank. So the cell separates the copied chromosome to form full set. That is known as cytokinesis, and interphase. That is a period between cell division. The period between two M phase is known as interphase. Therefore, the period between cell division, two successive cell division, it is known as what? Interphase. Clear with that? It is known as what? The interphase. Okay. So then we have to discuss about G0 phase. All those things we will do. That's why I haven't discussed the first point because it's about mitosis. Okay, Harshita. Okay, Akash. Akash, can you say from which group you are? Because I am seeing you for the first time. Okay, G1 phase or the gap 1 phase. That's the first thing we are gonna learn now. Everything will be made clear for you. So I'm ready to repeat for you how many times you need. So if it's not clear, please say to me. Okay? Comment me at the spot. So G1 phase. It is known as the gap 1 phase or the first growth phase. It is known as the gap 1 phase or the first growth phase. Okay. Or G1 is also known as generation 1. It is also known as generation 1. Clear with that? So, and look here in this figure, you can see a mitotic phase. Mitotic phase consisting of four subphases, that is the prophase, metaphase, anaphase and telophase. All those things you know, just have a base idea with that. Just understand these terms. Okay, this is the mitotic phase. This totally is known as the mitotic phase. So look here, a gap 1 phase, an S phase, and the gap 2 phase, and the mitotic phase. So that's how the cell cycle goes. So as you all know, the first growth phase or G1 phase occurs after mitosis, right? It occurs after mitosis. Therefore, it is known as the post-mitotic phase. The post-mitotic phase. Post-mitotic phase. Clear with that? Cell growth, continuous cell growth. Cell becomes metabolically active, prepares prepares machinery for DNA replication, synthesis, RNA, and proteins. So before moving on to that, let me explain some more things. So longest subphase of the interphase is G1 phase. G1 phase is the longest subphase of the interphase. It is also known as the gap 1 phase, generation 1, post-mitotic phase, or the first growth phase. Both the G1 and G2, both G1 and G2 is known as the preparatory phase. Both are known as the preparatory phase. High cell growth and metabolism occurs in case of this preparatory phases. High cell growth and metabolism. Okay, and what happens is synthesis of nucleotide, ATP, amino acid, RNA. Synthesis of what all occurs? Synthesis of RNA square occurs. It is the code for you to learn. Synthesis of RNA square occurs in case of this G1 phase. Code for you to learn. R refers to the RNA, the very basic RNA. N refers to the nucleotide. A square refers to ATP and amino acid. Therefore, we can say the synthesis of RNA square occurs. Do not write in the exam for that. Okay, it's a code for you all to learn because RNA synthesis occurs where, ATP synthesis occurs where, all those things are asked for the NEET examination. That's fine. Okay, I have given such a code. Clear all about this G1 phase. This is all about the G1 phase. It is the post-mitotic phase, that is occurs after M phase. Clear? Yes, adenosine triphosphate is known as ATP, Harshita. Good. Next is the S phase. So it is the longest. No, that's wrong. Okay, that's wrong. The sound from the temple is hearing in between. That's why. Okay. The longest subphase of interphase is G1 phase. Okay, it is not S phase. So just corrected. The longest phase of interphase it is G1 phase. Okay. And in case of S phase, the main event, the hallmark event is the DNA replication takes place. The DNA replication takes place in hallmark event is DNA replication takes place in S phase. Clear with that? amount of DNA per cell doubles, but chromosome number is not increased. So, in case of S phase, what happens is that the DNA number of DNA becomes twice. That is the content, the chromosomal content changes. That is, the normal chromosomal content is referred to as 2C. 2C. C refers to the content of chromosome. Okay. So what happens in case of this S phase is the chromosomal content doubles and it forms 4C. Okay, C refers to what? It refers to the content of chromosome. Okay, so that is what meant by the DNA replication occurs. In animal cell, replication begins in the nucleus and centriole duplicates in the cytoplasm. All of you know that, or you're gonna learn all those things when you're gonna start with mitosis. Clear? That's all for the S phase. That's all for the S phase. Okay, and one more event occurs in S phase. One more event occurs in S phase. That is the synthesis of histone proteins. All of you know histone proteins. Guys, comment me whether you know the histone proteins, whether you have heard about this. Have you ever heard about histone proteins? In order to avoid the sound of the temple to get rise, I am continuously speaking. So just understand that. Okay. Or else I would have waited for you. But if I am not talking, what happens is that the temple from the sound from the temple get rise. Okay, the histone proteins refers to the basic proteins or the basic amino acids present in what? The DNA, right? So the what? Double helical structure. All of you know the double helical structure, right? Oh my god, the structure get changed. Awesome. Okay, the double helical structure. If we take a chromatid and just unwind it, you can see an histone octamer. That is 8 histone proteins arranged. 8 histone proteins arranged. That's what about histone proteins. And so, such a synthesis of histone protein occurs in S phase of interphase. Clear with that? Then G2 phase or the gap 2 phase, also known as the generation 2, and it occurs just before the mitotic phase. Okay, it occurs just before the mitotic phase, and it is known as the pre-mitotic phase. Guys, now comment me, what does G1 phase known as? Since it is occurring after mitosis, what G1 phase is known as? Comment me right now. First, second growth phase is known as this G2 phase. Cell growth continues, and what happens? The hallmark event is synthesis of more RNA and proteins. Okay, not synthesis of RNA and proteins. Synthesis of RNA and proteins occur in G1 phase. So, synthesis of more RNA and proteins. Yes, Harshida, post mitotic phase. Harshida is too active in this session. Nila getting too slow. Nila getting too slow. Calvin, you are also coming up, but Nila is going too down. Be active, Nila. Okay. So cell is prepared for mitosis. In case of this, don't take it bad. I have just given an advice. Be active in the class. Okay. Like Harshida and Calvin. In most of the class, I have seen Nila being active. So just leave all those things. Okay. Just focus on our class. Okay, then this is all about G2 phase also. Okay, so all of you know how this mitosis occurs. So how the cell division occurs. How the cell division occurs. Cell division occurs, that is G1 phase, S phase, G2 phase, and M phase. And before this, before coming on to the slide, You all have heard a term known as the G0 phase. Now we have to learn what is this G0 phase, right? We all have learned about this G0 phase. Now what is this G0 phase? So before moving on to that, let me move on to the next page, so that you have a slide there waiting for you. Okay, look here. Hope this is perfect and fine. So look, the G1 phase, S phase, and the G2 phase. Then M phase. This is known as the M phase, right? Okay. Now listen. This is how the, yes, Arshida. That's how the what? That's how cell cycle occurs. So now you have to. Now we are going to introduce a term known as the G0 phase. So the cell prepares to its division and passes on to the G1 phase. And while the cell is moving on to the G1 GS phase, very sorry, I'm getting a little bit confused with that while speaking. Okay, just leave all those things. Just focus from beginning on this slide. Okay, wait 1 second. So the cell moves from G1 phase to G2 phase. In between that, there is a phase known as the G0 phase. In between that, there is a phase known as the G0 phase. That is, it is a decision taking place. Decision taking phase. Decision taking phase. Okay. So decision taking phase is the G0 phase. That is, decision of a cell whether to undergo further cell cycle or not is being taken in this G0 phase. Clear with that? So cells are metabolically active there, but they do not divide. So what happens if you are not clear along, if again it's not clear with that, what happens about this G0 phase? Now itself I will make it clear. 1 second. Let me take the screen. Okay. Yes, question phase. Exactly, question phase. Okay. G1 phase, S phase, G2 phase, M phase. This is how cell cycle goes, right? Is it clear till now? This is how cell cycle goes. So while the cell is moving from G1 phase to S phase, there is an additional phase known as the G0 phase. This is how you draw the figure. Okay. Exactly, Harshida. Exactly, like cardiac cells, nerve cells, and certain muscle cells, they too permanently remain at G0 phase. They do not go undergo cell division. Okay, they remain permanently remain at the G0 phase. Okay, that's how the cardiac cells, nerve cells do not get replaced. Once brain damage occurs, it cannot be cured. Right. So the cell passes from G1 phase to S phase. So in this between this, we have a checkpoint. We have a point, not checkpoint. Just leave that term checkpoint. There is a phase known as the G0 phase. So G0 phase, what happens is that the cell enters the G0 phase. The cells checks whether I have the capability to undergo the entire cell division or not, whether I can go all these stages or not, whether could I complete this or whether I should leave now, whether I should stop my work now. That is how the cell thinks when it enters the G0 phase. And if a cell if a cell after entering G0 phase understand that it can undergo further division, it moves back to the G1 phase and continues its cell division, that is to S phase, G2 phase, and to M phase, and finally next cell division, that is next cell division starting from G1 phase. Right? Is it clear? My God, my throat has gone. 4:51, but no topics have been covered till now. Okay, just leave all those things and focus on what we are gonna learn. Is it clear about G0 phase now? Is it clear about G0 phase now? Yes, okay, fine. So there are three checkpoints. Now I have used the term known as checkpoint, right? So there are three checkpoints. One is the cell growth checkpoint. Second one is the DNA synthesis checkpoint. The DNA synthesis checkpoint. And third one is the mitosis checkpoint. So just understand that the cell consists of three checkpoints in order to ensure that proper cell division is going on. In order to ensure the proper cell division. Okay, it checks the capability of a cell to move to the next phase. Checks the capability of the cell to move to the next phase. Clear with that? That's all about cell cycle. Just don't be confused with all other points. That's the thing that you have to learn here. You have all these things today. My God. Okay then guys, can we move on to the M phase? Again the screen is blurred. Oh my God. What to do with this network? Just let me refresh. 1 second. Is it clear now? I hope it's clear now. And when I'm gonna start writing, yeah, exactly. That's the point. When I'm starting writing, it is getting blurred. So I'm not gonna write now. Just understand, and if it is there anything important to write, we will do it. Okay. So that's all about the interface. And if you want in detail, surely I will do it. But today I have a limited time, right? We have a limited time because or else your time itself will be going. I'm free till 6 o'clock. Okay, I'm free till then, but I cannot take your much time. That's why. So cell cycle and cell division. It occurs in somatic cells. Oh my God! Again I forgot and I just marked it. Very sorry for that. And we just adjust it. Okay. So mitosis, what you learn about this mitosis, we can see mitosis is known as the equational division. Equational division. That is the number of chromosomes present in the parent and the progeny cells is same here in case of this mitosis. Number of chromosomes present is same in case of the both this in case of this mitotic division. Okay, so it occurs in somatic cells and in case of gametes, what type of cell division occurs, guys? Comment me right now. What is the type of cell division occurring in gametes? I'm moving very slowly. Hope it's very clear. Okay, I just plan to finish it in 2 sessions. Then only I will be able to complete it in at least 4 sessions. Okay, that's fine. And remember, if I am not able to take live, surely premiering will be done. Okay, but I am not getting time for both. What can I do? Let me just be see. I have some works there. Okay, that's fine. Yes, Harshida. Meiosis is the gametes, what cells division of the gametes. Okay, just leave all those things and now focus on our topic of the M phase. And we have this Mitotic phase including four stages. Mitotic phase including four phase stages. That is a prophase, a metaphase, anaphase, and telophase. Very good. My God. Raining started here. Yes, Dima, you're back. I don't know whether it will be disturbing you or not. I don't know. When I am coming, surely there will be some issues for me to take the class. Please just adjust if you are not able to hear properly. Just tell me. So four stages occurs. That is the prophase, metaphase, anaphase, and telophase, and we are going to discuss all these things in detail now. Clear with that? So without wasting much time, let me move forward. If I'm moving fast, surely you can say. Okay. So the first phase, that is the prophase. First phase of mitotic phase, that is the prophase. You can see a GIF here. You can see the GIF here, right? The cell division till anaphase and what telophase and cytokinesis itself is shown here in the GIF, right? Okay, okay, Harshida. So just next we have to learn what we have to learn in case of this chapter is entirely based on theory part, and we have bit fun while drawing the diagrams. Okay, that's the only thing that you have to learn, diagram. Okay, that's only interesting from this chapter. All other parts are theory. Okay, so now we are going to learn about the events of this different stages of mitosis. What all are the things happening in prophase? What all are the things happening in metaphase, anaphase, telophase? All those things that we are gonna learn now. Okay. Oh my God, my throat is gone, shouting and shouting. In order to avoid the sound from the temple, I am raising this much voice. Okay, then let's focus. So what happens is the condensation of chromosomes occurs. That is the chromosomes. As you all know, it is just fully what crossed inside the nucleus, right? So condensation occurs. That is the chromosomes get starts to get duplicated. That is like this. Okay. So condensation of chromosome occurs and spindle apparatus assembles. So if you check in this GIF, if you check here in this GIF, you can see this spindle fiber formation, right? Spindle fiber formation, right? And the centrioles. Look, the centrioles. One second. Look here. The centrioles moving to the poles. The centrioles moving apart to the poles. Asters or radial ray of microtubules. That is not in animals, not in plants. In plants, there is no centrioles. As you all know, no centrioles is there. Yes, Arshida, chromatin becomes chromosome. Okay, astral rays are present. Radial astral rays of microtubules are present in animals and not in plants. Okay, because centrioles are absent in plants and just spindle fibers are just seen in plants. And the nuclear membrane breaks up. Look here, the nuclear membrane breaks up. Excuse me. Nuclear envelope breakdown. That's what are the events occurring in prophase of mitosis. My God, extremely horrible with shouting. Okay, so look here in this diff, you can see the mitotic spindle beginning to form, the centrioles moving to the poles. Okay, and the chromosome condensation of the chromosome, the nuclear envelope breaking down. Okay, all those things you can see in this GIF, right? Exactly, Harshida. They also degenerate. That all the cell organelles degenerate and replicate. All those things occur in the S phase. Okay, just mark it. In the S phase, all those things happens. Endoplasmic reticulum, Golgi bodies, and all those things just degenerate in case of S phase. Okay. So is it clear about the cell cycle, mitotic phase, prophase of this mitotic phase? Endoplasmic reticulum, nucleolus, nuclear membrane, everything disappears when they all replicate in case of a space. As I have said, they all replicate in case of this space. They all disappear in case of this late prophase. Okay. Okay. Fine. So now we are moving on to metaphase. Look, this just you can see all the chromosome, the hallmark event and the basic event occurring is the chromosomal align. Chromosomes align at the metaphase plate. They align at an equatorial plate that is exactly at the center, and that is known as the metaphase plate or the equatorial plate. Okay, so chromosomes align at the metaphase plate in case of this metaphase. Clear with that? All these figures you can see it very clearly. Even in this GIF also, it's very clearly depicted. Is there any doubt still now? Is there any doubts? And one more thing, if a question asks, which is the best stage to study the morphology of chromosome, it is the metaphase. Okay, so the to study the morphology of chromosome, it is the metaphase because it is the perfect stage having the what all chromosome aligning at the center and with the perfect structure of the chromosome. Okay, okay then. Next we have the anaphase. Here another GIF is given. Okay, another two GIF are given. Look, so the chromosome, we all have seen that in the metaphase plate we have aligned this chromosome, right? All the chromosome align equally in a metaphase plate. That's what we have learned. So that what happens is, as you all see, they get splitted from the centromere. So centromere splitting of centromere, excuse me, splitting of centromere occurs. Okay, splitting of centromere occurs in case of this anaphase. Okay, and chromosomes look like the chromosome formed look like J-shaped, L-shaped, V-shaped, and I-shaped chromosome. All these have different names. The centromere at the end, terminal centromere, middle centromere, all those things known as acentric, all those things. Okay, telocentric, all those things. Okay, metacentric, submetacentric. Okay, submetacentric chromosomes. These are the things. And best stage to study this morphology of chromosome, it is metaphase. Best stage to study the structure of chromosome, it is anaphase. Morphology, it is metaphase. Structure, it is anaphase. Clear with all these things till now? Is the class useful or I am wasting your time? Am I wasting all of your time simply by talking? Okay, fine. Yes, Harshida, V-shaped is metacentric. T-shaped, that is somewhat I like this. Like this. I don't know whether you can see or not. No, it is not at all visible. I hope it's not at all visible. So very sorry for that. Okay. Oh my god, it's another thing. If I get this network in my hand, surely I will kill that. like this. This is how what is meant by the I-shaped chromosome. Okay, and the L-shaped chromosome, if we say, it will be like this. V-shaped chromosome, it will be exactly in the middle. Yes, yes, yes. I-shaped it is metallocentric. Exactly. J-shaped it is acrocentric, not acromia, acrocentric. Okay. Guys, you all know everything. Then why are you making me to repeat it again and again? My God. Yeah, just move on to the telophase. What happens is that the spindle fiber starts disappearing. Disappearing of spindle fibers occurs. Nuclear envelope forms around. Nuclear envelope reappears. Chromosome begin to decondense. Okay, decondensation of chromosome. That is the duplicated chromosome formed like this. Like so, they again begins to coiled. Okay, clear? My goodness. Next is cytokinesis. Is it over? Yeah, fine. Yeah, very good. Okay. Cytokinesis. That is the last stage of the mitotic phase. Okay. Yes, exactly, Harshita. Back to chromatin. So, in case of cytokinesis, what happens in case of animal cell and plant cell? Animal cell, what happens? A furrow forms in between. A furrow forms. As you all see, look here, a furrow forms. Can you all see this? And in case of plants, a cell plate forms. That is centrifugal in direction. That is centrifugal. Okay, and in case of furrow formation, it is centripetal, that is towards the center. And the cell plate formation is centrifugal, that is towards outside, from center to outside, that is cell plate formation. Okay, so cleavage of the cell into equal halves. Cell division occurs into two equal halves, and division of cytoplasm is known as, resulting in the formation of two daughter cells, two identical daughter cells is known as cytokinesis. Is it clear? So look this GIF. Prophase, metaphase, anaphase, and telophase is demonstrated in this GIF. Cytokinesis. Look here, the cell divides and two daughter cells are formed. Is it clear? Was I a bit fast and have you understood all those things or not? I hope I felt I was a bit fast and but I have covered all the points. All the points which I have to say, I have covered it. Very important points I have covered. Okay, least and very important points I have covered. That all, that only those things are needed for you. And other points which you have to learn from this, you will learn in meiosis too. Meiosis too is same as mitosis. Okay, Harshida, Dima, Calvin, Nila. Very sorry, Nila, since your name is getting too small today. Okay, because all others name big name. Dr. Dima Divagaran, Rajni Bala Chituri, Calvin Christopher. But from that, I was what I'm difficult to see your name. That's why. Okay, I haven't forgot you. Okay. So significance of mitosis. Significance of mitosis. What we say in case of that is produce daughter cells with identical genome. Genetic material is identical. Retain same chromosomal number in all somatic cells. The now chromosomal number is same. That is a hallmark point. Maintain normal chromosomal number. Retains the nucleocytoplasmic ratio that disturbed due to cell growth. We have said that the chromosomal content changes in case of this S phase. So after mitosis, what happens? It just gets restored. Okay, and it helps in repair and replacement. Clear with that? Clear with that? This is all about mitosis, and now you have a video waiting for you. A video waiting for you. I don't know whether you will be able to hear the sound or not of this video. Anyway, just wait and see. Okay, just wait and see all these things. Okay, So I'm gonna play this.
During interphase of the cell cycle, the genetic material of the cell is found in the form of chromatin and located within the nucleus of the cell, which is surrounded by the nuclear envelope. During the S stage of interphase, DNA replication takes place, and thus the cell leaving interphase and entering mitosis already has replicated its genetic information. In addition to the replicated genetic information, interphase cells that are about to divide also replicate their centrosomes. In animal cells, these centrosomes are formed from centriole pairs. As cells leave interphase and begin prophase, the first stage of mitosis, the chromosomes begin to condense. The nuclear envelope begins to vesiculate, and the centrosomes migrate to opposite ends of the cells to define the two poles. During prophase,
Look now, you can see the centrioles moving towards the poles, right? Now you can see the moving of the centrioles towards the poles. Okay.
the mitotic spindle apparatus begins to form by the polymerization of tubulin proteins. This creates microtubules that emanate from each centrosome. Chromosome.
Oh my god. Oh, very sorry, guys. Very sorry. Very sorry, it's not getting playing now. Off. Just wait for a bit. I feel it will be all right now. Just wait. This video is the end for the day. But anyway, is the topic total now clear? Is the topics clear? Both interphase and mitotic phase. It's the issue with the PowerPoint, and I was a part of that. I just changed the slide and okay, 1 second. Oh my god, PowerPoint is stopped. 1 second. Please wait. 1 second. Soon it will be back. I'm taking time for this PowerPoint to get restarted. Okay, that's right. Very sorry if I'm wasting your time. Till then, have a break to drink water. Okay. Okay, come on fast. I'm waiting for this PowerPoint. This PowerPoint is making you all wait. Yes, okay, Harshida. Okay, Calvin, Nila, Dima. Everyone, kindly cooperate. Okay, it's by mistake. Yes, it's get opening. Please wait. Processed and opened. Finally, we succeeded. Guys, we succeeded. Oh my god, before that. Again, it's hang. My god. Please wait.
During interphase of the cell.
1 second, 1 second, 1 second. 1 second. It's starting by its own wish without asking me. Surely it's fun this class. Can you see the screen? Can you see the screen? Is it clear? Okay, fine. Oh my god, again.
During interphase of the cell, find the two poles. During prophase, the mitotic spindle apparatus begins to form by the polymerization of tubulin proteins. This creates microtubules that emanate from each centrosome. Chromosomes completely condense, such that sister chromatids formed from the replicated genetic information enjoy that their centromeres are clearly visible. During prometaphase, the nuclear envelope completely breaks down and the mitotic spindle is formed. Polar microtubules extend from both poles and overlap with each other. The sister chromatids become attached to the mitotic spindle via kinetochore microtubules that attach to the kinetochores, which are bound to the centromeres. As prometaphase is nearing its end, the sister chromatids are observed to jerk back and forth between the two poles. During metaphase, the sister chromatids become localized in a region called the metaphase plate. Once the chromosomes have aligned at the metaphase plate, metaphase is complete. When metaphase is completed, anaphase begins. The sister chromatids separate, and each chromatid is now linked to only one pole via a kinetochore microtubule. The kinetochore microtubules shorten, and the individual chromatids, now called chromosomes, are drawn to the poles. Toward the end of anaphase, the polar microtubules push against each other and cause the two poles to be moved farther away from each other. Each daughter cell will receive the same complement of chromosomes as was originally found in the mother cell, thus assuring that each daughter cell gets an identical copy of genetic information. During telophase, the chromosomes decondense, and most of the microtubules depolymerize. Portions of nuclear membrane surround each individual chromosome, creating vesicles. The vesicles fuse with each other to create a nucleus containing all of the chromosomes at one pole of the cell. In animal cells, cytokinesis or cell division involves the formation of a cleavage furrow. The plasma membrane is observed to constrict and eventually separates the cell into two daughter cells. The daughter cells then enter interphase, where growth and enlargement occurs, leading to either preparation for another mitosis or differentiation and specialization occurs.
Okay, guys, clear with that? And that's all for today. Do not forget to hit the like button. Do share it with your friends. And if you are a new member, do not forget to subscribe the YouTube channel by Point Stream the World of Bio. Today's class was a bit fun. Okay. And this last video had the entire part of this today's class, both interphase and mitotic phase. Okay. So thank you for your wholehearted cooperation. A special thanks to all you bought, Nila, Calvin, Dima, and Harshita. Yes, bye. With more fun, I will be there before you in the next class too. Okay, a fun loaded class. Okay, then, guys, it's me living from your favorite and my favorite. Bio Point Stream the World of Bio. Bye bye. Just remember one thing, I haven't made fun. It's a fun made by the network issue. Okay, I'm living. Very sorry for that. Bye, guys. Take care. It's me living from your favorite and my favorite biopoint. Today we had 1 hour class. Very sorry for taking your precious 1 hour. Okay. You all may leave or else I will keep on talking. Okay. Once I started, I won't end it like that. Anyway, bye guys.